So yeah, I'd like to talk today about how we do engineering in CMap and specifically around our data processing pipelines. We've got several other worlds of engineering involved with our front-end apps, our Clue website. I'm not really going to touch on that. That hasn't really been my area of focus, but there's a lot of amazing work that goes on there as well. And also some of our friends, which I'll get into what I mean by that in a little bit, related groups and collaborators who do similar things, and we work with to varying degrees. So a brief outline of the talk. I'd like to first go over, give you some background about CMap and who our friends are. I mean, we're all friends here, but this is sort of my euphemism for people we work fairly tightly with who do similar things. Then I'll talk about some of the challenges we face, some of the engineering challenges and sort of how they tie into what we do and how we go about trying to tackle those. And then finally, I'll show you some of the results, some of the tools we've built over the course of the last year and a half. So CMap, the short version is it's the connectivity map. Many of you have probably heard of that. For those who don't know, that's what CMap stands for. The long version is that we're trying to, collectively CMap, we're trying to build a library of perturbational signatures. So what does that mean? I'll get into what we mean by a signature in a little bit, but the library part, we want it to be comprehensive, like the Library of Congress. We'd like it to be information rich, so not optimized to answer one specific biological question or scientific question versus another. It should be easy to use, so you should be able to look things up rather easily, like Google. It should be easy to use non-CMap data. You shouldn't sort of have this sort of lock-in that only if you run CMap or our specific L1000 assay you can use it. It should be general to lots of other data types. And it should be accessible to both lab and computational biologists. So the experiment that we do that generates a signature is pretty straightforward. We perturb some cells. So we have some cells generally in a plate, and we apply some type of perturbagen. The main ones we do are shRNA, small molecules, CRISPR. We do some overexpression constructs. And then we measure a phenotype of what happened as a result of applying that perturbagen. And the cell lines we use, we focused on 8 core cancer cell lines for the vast majority of our experiments, but we have branched out quite a bit into other types of cell lines. And the phenotype here is each of these lines actually represents an individual experiment, and so it's a multiplexed assay. So these bars represent individual measurements within that multiplexed assay. So that's one of the key features of what we're doing and what our friends do. And I just want to take a moment to mention, none of this is possible without amazing work by the folks in the lab who do really diligent experiments, very carefully, very methodical. None of what I'm going to talk about would matter if we didn't have them working hard in the lab. So the phenotypes that we're measuring, the multiplexed measurements we're making, for CMap, the core one is really the L1000, it's gene expression. Some of the friends that I've mentioned earlier that we're working closely with, one is measuring viability via PRISM, another is proteomics measurements via P100 assay and global chromatin profiling, GCP, and then imaging via the cell painting assay. And again, the common theme is all of these are multiplex. All of them have a decent number of readouts for each single experiment. And the collaborators there's a lot of people in each of these groups, both in the lab and in the computational side. I just want to highlight a few that we work the most closely with. Viability, Jordan Bryan, Aviad Cherniak, Chris Mader for PRISM. Proteomics, Ryan Peckner and Jake Jaffe, and then imaging, Shantanu Singh and Ann Carpenter, and of course many others behind the scenes that I haven't mentioned. And I'd like to give you an example of really what we mean how we think about our multiplexed assay data. It's basically a matrix. And what we look at is, so the columns we think of as different experiments we've conducted. So in this case, I've labeled the columns of this matrix. The matrix is represented as this heat map. I represent the columns as what perturbagen, what cell line we used and what perturbagen we applied. So in this case, I'm just showing you these are the compounds we applied for the experiment, and then this is the cell line. In this case, it's all A375. And then the rows are the different measurements we make in our multiplexed assay. So in this case, it's the genes we measure. We're measuring the gene expression level of these different genes. Or in this data, actually, it's the differential gene expression. So it's the gene expression with respect to the control. So for our friends, you know, what they're doing is it's the same idea. We think of the columns as the different experiments that were conducted, and the rows are different measurements. So in PRISM, in the case of PRISM, it's the viability of a cell line. So in a single well on a plate, they can measure 25 to 30 cell lines viability simultaneously. That's a very powerful technique. And so they get a readout where, in this case, actually the cell line, they have a range of cell lines here, and then these are the conditions that were used to treat that pool of cell lines. For P100, the row readout is the amount of phosphorylation of site X. For global chromatin profiling, it's the amount of histone modification that happened on a set of sites. And then finally, for cell painting, it's the amount of observation of some feature Z that they've measured and analyzed using cell profiler.
So sorry. For,
sure, please.
Viability is, how is viability in multiplex readout?
Well, the reason it's multiplexed is that in a given experimental condition, they'll have a pool of cell lines, 25 cell lines. And they'll have those 25 cell lines all together in a single well, and then they'll measure that. And then we actually, that's how they prepare the lysate, is in pools of 25 cell lines. And then we actually, after we've prepared the lysate, we can pool that down into, so we effectively have the lysate from 300 cell lines that have been treated all in the same conditions in a single well for detection. Thanks. Good question. So just to dive into a little more detail for L1000 and our gene expression, this is our data processing pipeline. It's called Espresso. I'm going to sort of review the data levels here at a high level. We have analogous data levels for the other multiplexed assay, and that comes in really handy later on for usability, for allowing analysts to look at results from multiple assays at the same time. Having the data levels aligned and the metadata aligned is sort of key for that. Otherwise, you can end up spending a lot of your time doing that rather than doing analysis. And I'd like to mention also, like, this pipeline was built mostly by Rajiv and Ted. And I think did Arvind help also, Ted? Okay. Okay, I thought so. So yeah, and I've just sort of come along and done some engineering work on top of this, but the bulk of the really heavy lifting was done by these three. So the data process starts with level 1 data. These are LXBs. They're binary files that come off the scanner when we've made the raw measurement in the lab. The first thing we do is we do a deconvolution experiment because our scanners actually measure 500 different analytes directly, but we want to measure 1,000 different genes, so we have a process for deconvoluting those 500 colors into 1,000 measurements. That's the first step. That sort of gives us a very raw gene expression level. We then do some normalization process to controls on the plate and other controls in each well to give us normalized data. We then do inference because while measuring 1,000 genes in a single shot is great, especially for the cost and the speed with which we're able to do it, most people are interested in, there's an additional 21,000 genes in the genome, and it would be really a problem if what an external person was interested in didn't happen to line up with our 1,000 measured genes. There goes your usability and accessibility, one of the sort of main goals of the library. So we have an inference model which can infer or extrapolate from our 1,000 measured genes to an additional 21,000 features. Finally, after that, we do differential gene expression. So that compares the gene expression value we've measured in the cell to negative controls, getting, again, to what was the effect of the perturbagen that was applied. And then finally, the last part of this pipeline is aggregating the individual signatures together, aggregating the replicates together to get a higher signal-to-noise signature. This is a little out of scope, but I think it's important to mention, going along with our coffee theme, we have another pipeline called Macchiato. So if anyone's familiar with Macchiato, is an additional feature you can add to an espresso drink, if you will. This is running in the cloud. Jacob and Rajiv have ported this to work on AWS, and basically what it does is it takes the data that at the end of the Espresso pipeline and really gets it into that final state where it's easy to look up and run queries against. And that's an important part. And there's a lot of engineering that goes into that as well that I'm not going to talk about today. So that's sort of a quick overview of, wow, that's almost invisible. That was a quick overview of what CMap is and who our friends are and sort of what we're working on. Now I'd like to talk about the challenges we face and how we've gone about tackling them. So, and I'd also like to talk about how these challenges relate to the ilities. If you read the abstract, I mentioned the sort of engineering ilities, and these are sort of pretty standard for a lot of software engineering. So, first one is scalability. We do need to be able to scale up our capacity. In the time I've sort of been here, I think our lab capacity has gone up about threefold, maybe a little more than that. And we needed to have our pipeline be able to scale up and still process the data in a timely manner. Usability, again, generally we're not talking about end users, biologists, and making applications that are usable for them, but we do have some usability issues in that we do a lot of quality control. We generate a lot of graphs and plots and diagrams for lab to give them feedback on how the processes are running in lab, and it's important for those to be usable by the lab folk. We have scripts we use for analysis. These are a little more technical, but it's equally important that any analyst on our team should be able to go in and, without too much pain, be able to fire up one of our scripts and be able to use that. And then finally, as I mentioned earlier, data alignment. We want to make sure the data levels across all these assays are relatively synchronized and that the metadata isn't too disparate. And that's always a challenge and an ongoing challenge. So then maintainability, we want to be able to fix bugs in a timely manner to the core pipeline, and we want to be able to make modifications as needed to the pipeline. And that's a general software challenge. We've got that challenge just like everyone else. In terms of extensibility, one of the big ones we've focused on is adding new QC modules, new ways of checking the quality of the data we're producing. And also we've worked on adding new pipelines. So we've worked on building part of a pipeline for the PRISM assay, and certainly Lev has done a lot of work, we'll mention this in a little bit, on building a pipeline for the P100 and GCP assays. And then finally, portability. We want to be able to allow collaborators and others to reuse our code. So the solution that we've been doing has been trying to use engineering practices, some reasonable set of engineering practices, and then bringing in awesome computational biologists to use those engineering practices to make it all happen. So the engineering practices at a high level make it possible for us to address new and unforeseen challenges as they come up. So initially, the engineering practices were pretty simple. Just for me, working on the system, it was improve the logging so I can sort of understand what's happening with the system in production. As problems arise, it's easy for me to see when it happened and what modules it happened in. And then, thanks to Phil, this became my mantra. Whenever you see cron, whenever you have a job that needs to run repeatedly, instead of using cron on Linux, use Jenkins. It's a great software package for that kind of thing. And then starting to add tests. Now this was all great. It helped me out. It made my life a lot easier, starting to follow these practices internally. But that only took us so far. That allowed me to sort of keep up with the production pipeline as capacity increased. It really didn't solve the problem of adding new QC modules or adding a lot of new features. And that's where we decided it was time to release the ACBs. And so, I had the pleasure of working with all of these associate computational biologists and computational biologists in the mix, and I'll talk about their work individually, but I just want to give them credit because they're really the ones who've done the bulk of the work that you're going to see here today. So the benefits of the associate computational biologists, it's a little obvious, but they're going to vastly expand the capability to add features, add new modules, things like that. Almost equally important is they provide a fresh perspective and new ideas. That's invaluable. It's very easy, I know I do this, it's very easy to get stuck in our ways and sort of only think about things from one perspective. When you have new people reviewing your ideas, challenging on your ideas, it's invaluable for questioning your assumptions, reevaluating them, and finding mistakes you've made. What the ACBs gain from all of this, and this is also, their gain is our gain, I think, in the sense, they get knowledge of existing tools, the algorithms, the workflows, where to find data that they'll need when they go on and do additional work in the future. And they get some exposure to engineering. That may or may not help them in the long term in their career path, but most of them may or probably won't become engineers. I don't think that's the goal here, but I think having some exposure to engineering processes and discipline is useful for anything you do. What are the risks? What could possibly go wrong? So to illustrate this, I'll give you an anecdote from right when I started as a software engineer. The first project I worked on was the first project of everyone who joined the company. And the company I was working for didn't hire software engineers. They only hired scientists who knew some programming. So there was no engineering training. There was really hardly even any computer science training of all of us. We were just sort of, hey, I think the logic was roughly that you're not ready to be client facing. We need you to sort of do some work though, so work on this thing. And the result that everyone just sort of dove into it. Everyone's working hard, they're trying their best, but still, without that experience or any of that training or that overview, everyone sort of came up with a new way of doing things and just wrote that code and just jammed it in there and stitched it in and made it work. So there were many, many redundant modules, classes, functions, and data structures. And I think it might have taken me 2 weeks or even a month, I forget. I wasn't on the project that long, but I started to realize, wow, all the parallel data structures. It was just amazing to realize, I was working through one section, and then I went to another section, I was like, wait, these do the same thing. Why are they both here? And then it sort of dawned on me what was sort of going on. So I think a good word for that is spaghettification. You sort of end up with spaghetti code, although I would think it's both in the code and I think also in the philosophy or the architecture of the system. You can just end up with everyone doing their own thing. So I think I sort of bring this up to say, okay, if we sort of just naively said, okay, just without any guidance or any instruction, we just turned a group of scientists loose on the code, that's what could happen. And I think that's a risk. So how do we avoid that? By being lazy and stupid. I think that is a great way to summarize the motivation for why we do a lot of the engineering practices we do. And I'll get back to you. I'll explain that a little more in a little bit. So some more specific description of what we do. We try to provide training to people, provide frequent feedback, and by training I mean mainly hands-on training, frequent feedback on how people are writing their code and what they're working on, writing tests, automated tests, documenting the code and writing self-documenting code, and then of course iterating all these processes to trying to keep track of what we like, what we don't like, and adapting as we go. And then finally, documenting our institutional knowledge of what our best practices are, what we think works, what doesn't work, writing introductory guides for people who are new to the group to sort of get them up and running as fast as possible. So I'll now sort of go into some more details of these. For the training, some of the things that I think have worked well is when we try to work one-on-one to design and plan out our modules before we start coding. We've done this to varying degrees depending on the project, but we try to write a little short spec about what we're trying to do to make sure we're all in agreement, we all understand what the project actually should be. You think you can, I mean, in the company I worked for before, the spec could become a really big, monstrous thing with a lot of sections and a lot of documentation. I tried to avoid that here and just get the ideas down on paper. It's almost just a way to sketch out your ideas of what you're building before you actually build it. And then another one is sort of on-demand explanations or exposition about topic X. And Joe, one of our ACVs, came up with this central dogma of CMap. Rajiv has already done it. If you're getting tangled up in something, odds are Rajiv already solved the problem, and he probably wrote a pretty good function to do it that's pretty easy to use. So it's definitely worth going to talk to other people. And on the flip side of that is that we have the understanding that our senior engineers, our senior computational biologists, will make themselves available to people who have questions and take the time to explain topics as they come up as needed. And lately it's become go see Andrea, she knows SQL, or go see another ACV because they've become an expert in that field. So one of the things at the outset was, Arvind and I talked about when we started doing this was that, okay, well, hopefully, there may be a steep learning curve for all of us for the first generation of ACVs. Thank you, Lev, for suffering through that, being the very first. But then they will go on to then be the instructors for the next generation of ACVs. And of course, read the documents. So another thing for training is providing the time to learn. So I mentioned providing the time of the senior people in the group to give the instruction, but I think also it's really useful to really set aside time for individuals to go learn. So take, when someone has to learn SQL, take a day, take 2 days, read the online tutorials, set up a SQLite database, start creating tables, start messing around with that. As part of that, look at, once they sort of have that basic familiarity and they've had that time to sort of play around with it, they can go start to look at the code we already have in use. Take that time to, not be in a rush to produce something right away, but instead pause, learn the technology, at least get the beginning familiarity with it before diving in. And then finally, pair programming. Pair programming is, I think it's fantastic. If I had more time, I'd spend a lot more time pair programming with all the ACBs. I feel like it's incredibly useful for both. It's almost like a picture's worth 1,000 words. It's like actually sitting down and looking at with someone what they're trying to do is so much easier than standing in the hallway trying to explain what you're trying to do, or I'm trying to use this library to do this. Well, just show me. Let's look at it. Let's walk through it. Let's try to debug it together. And similarly, explaining the explanation can be a lot more fruitful that way too, I find. In terms of feedback, we do code reviews twice a week for 30 minutes. It's not a lot of time, but it does give us a flavor of what's going on. And I particularly really like that people come to code review with things they want feedback on. They've figured out like, oh, this is bothering me. I'm not sure how to put this together, or I've got this working, but it seems like it's kind of clunky. It's really great that people come eager to get feedback on their code.
Question about code review.
Yes.
The interruptions there.
Yeah.
Do you do that in person as a meeting, or have you experimented with doing code review online, sort of on demand, as opposed to as a separate meeting?
Interesting. No, yeah, we have in-person meetings. I hadn't thought about that. That would be interesting. You mean kind of like almost like a really fast pull request. And
It's a question because I don't do that either. But
oh, okay.
It's a lot of thoughts. Code review creates some social interactions in person that might be different if it's offline.
Yeah.
And also it might be more efficient because then you don't have to have a meeting.
Yeah. That's interesting. Yeah, I'll definitely think about that. That could be useful. Does anyone know of any technologies you could use to do that quickly?
GitHub.
Just GitHub, yeah. Okay, great, thanks. So I think the other advantage of code review, besides feedback on the code or the structure of the code, is you get to learn what others are doing, and that helps keep you and others dry. Dry is one of my favorite acronyms for don't repeat yourself. I think I originally learned about it in terms of documentation, like you don't want to have documentation for the same piece of software in multiple locations, but I think it also applies to code and libraries. Like you don't want to rewrite libraries, rewrite code. Again, like if you want to, I think that's a great way to build up a knowledge of what code is out there is if you see what other people are writing and what other libraries they're using. You sort of can build up this internal catalog of what's available to you. Pull requests, again, as we talked about a little bit here, I hadn't thought about it for sort of the quick feedback. These usually are for deeper dives into the code. Ideally, I would like to review every line of code at least once for an ACB to sort of give them a very deep, in-depth overview of sort of my philosophy on how to write it, good or bad, take it or leave it in a way. But it's hard to do. That's very time consuming, and I will admit I have fallen behind many times on this, and it's a struggle. But I think it's very valuable. And then we have higher level sort of weekly or bi-weekly meetings, generally with some of the product owners or the project leaders to sort of review the progress as we're going. And then finally, we have office hours as needed. That is scheduled as a standing meeting, but it's nice in that if no one really needs the help, we'll just sort of hang out and work. If we do need the help, we have the time blocked off so it won't be invaded by other things. So then tests. These are something I've become fairly passionate about. Some of my justifications or rationalizations is that you're basically doing the testing anyway, and everyone I know tries out their code as they write it. I haven't met anyone yet who just sort of writes and writes and writes and writes tons of code, and then at the end of the project runs it and it works and it's fine. You're gonna, one way or another, you're gonna try your code out and your methods out as you write them. And so my contention is just those are basically your automated, just automate those, just save those. Don't throw that work out. You figured out how to run just method X and you know the inputs right now, you're doing it. Just save that, write it down in an automated test. Why? Because you're just gonna make my life so much easier. The other part of this, and again, this is nothing new to me. This is all ideas I've stolen from other people about software engineering. If you're having a hard time figuring out how to test your method or test some aspect of your code, then that means it's probably unnecessarily complicated. It's probably a good indication that you could simplify it and save someone in the future a lot of pain and effort. And again, this is all obvious stuff about tests, I think, but it's good to reiterate, you know, when I show up later to track down a bug in the system, I'll immediately be able to run your code. I won't have to spend a lot of time figuring out what are the proper inputs, things like that. I can just immediately get your code up and running and step through it in a debugger or whatever I need to do. As a person who is not the original author, if I need to extend your code and extend your test coverage to cover a bug we've encountered, it's a lot easier to copy an existing test and modify it than to start from scratch. Or in my case, if you're the author but you haven't touched it in 6 months, it's almost like you're learning it from scratch. And again, obviously this is not just for bugs, it's also for new functionality. You know, the question that happens all the time, you know, I ask myself did my change break anything? When we initially started and we had very little test coverage, you know, it was really dicey to know if a change I was making was going to break some other functionality. It's much easier to assess that when you have the automated tests written. So then for the documentation, the self-documenting code, one thing, again, analogous to tests, if it's hard for someone to write out the name of a variable or a function, it's a good indication that something's wrong. It's like a code smell. There's something too complicated going on. You really should be able to rearrange things so that you can explain it to yourself in your self-documenting code, and someone else can read it and understand what you're trying to do. And again, think of the people who are coming after you. They will thank you profusely. So again, strive to be lazy and stupid. I think that's another way of putting what I've been saying is, I keep saying over and over again, make it easy for yourself the next time you have to do this. Make it easy for yourself for the next person who has to do this. I mean, you can think of that as being lazy. You could try, again, don't repeat yourself. You're reducing the amount of work you have to do. That's where the laziness comes from. The stupid part, make it easy for yourself intellectually to be able to understand it the next time you come to look at your code, or the next person who has to come look at your code. I have no doubt that, for any given set of code we've ever written, any one of us could go in and understand it. Given a reasonable amount of time, we could figure out what we're trying to do. But why do we want to spend our effort doing that rather than doing science or building something new? I think we could do it. I don't doubt anyone's abilities, but we might as well be as stupid as possible, as often as possible, so we can work on other more challenging things. And again, don't be afraid to ask questions. This has many, many benefits. The two main ones, again, I'll reiterate, are, you're going to learn something, and you're going to force the person you've asked the question of to give a good explanation and reevaluate their assumptions and think about things maybe in a new light. And again, the why for all of this is we want to fix bugs quickly. We want to create new and exciting features. We want to get out there and work on the science. So that's sort of the challenges we've faced and how we've addressed them. I like to spend a little bit of time going over some of the results. This is just a little bit. This is a brief bulleted list of some of the things people have built. We've got Lev has extended the proteomics pipeline. I'll go into these in a little more detail. I won't go into them here, but this is just a subset of the things we've built in the last year and a half. It's been really fantastic working with the awesome computational biologists to do these things. So first, I'll talk about the proteomics signature pipeline briefly. This was originally in R. It was fairly well written. The code was self-documenting. That was great, but they wanted to move it into Python and make it more portable. Those were some of the main goals, and then start to extend it and add on to stuff, which Lev has done. And he set it up so we can use Conda as the environment. So we've got our portability achievement unlocked with that, which was pretty great. And these are the data processing steps in that pipeline. You can sort of see it follows the pattern I showed you in the graphic earlier. So that has been fantastic. Another one we've worked on is pandasGEXpress. Oana's really led the charge on this one. The idea here is we've got GCT file format has been pretty well established for providing annotated matrix data. GCTX is a binary equivalent. We mainly use that internally. It's based on HDF5 format, and we also use, pandasGEXpress uses pandas and the H5Py library to sort of bring these two things together. We can delegate the data manipulation once you've loaded your file to pandas, and we can delegate the storage to H5Py in the case of the binary. And we avoid reinventing these wheels. I'd like to jump over really quickly as sort of just to illustrate some of the things here we've talked about. In terms of, I think, this is the public GitHub repository, L1K tools. But I really would like to illustrate, for example, the documentation. Oana's done a great job of adding this very comprehensive README file, which is fantastic for how to set up the system and use it. Certain specific cases you might be interested in, and that's fantastic. But just to take it even a step further, she's also set up a Jupyter notebook with the environment and walks you through, again, a lot of major use cases. And this is very extensive and shows you how you can use the library to do basic operations that you might find useful. And that'll be fantastic for the wider community that can use this library, as well as our own internal folk. So then another tool Mark built is called Litichevskiy. So this is a plate, an experiment we run like a litmus test in chemistry, where you it's a quick functional test of our assay to make sure that it's performing as we expect it to be. The short version is that we've got a plate of perturbagens that are shRNA, so they're knocking down a gene, and each gene we've chosen is one that we measured directly. So it's we've got 330 on a plate, and those 330 are within the subset of the 1,000 we measured directly. And what we do is we just ask the question, did the shRNA in this well knock down the gene it targeted more than all the other experiments on the plate? And if it did, then we say, okay, well then the assay is working functionally. We expect to see knockdown of a gene, and we're seeing knockdown of a gene. And so we had some various measures of that, but Mark really took this to the next level by providing a lot of new figures. And I want to just illustrate this heat map, for example, here. So this heat map, you might notice the really weird color scale here. Well, basically, like if something is this is the rank of the knockdown score across the entire plate. And so if the gene in this well worked, it should have a rank of 1. It should be the most knockdown. It should have that gene knocked down more than the rest of the plate. And so that's why we're really interested in almost having a binary color scale, where things are okay if they're green and they're blue if they're not. But so we didn't have this before, but we now have this plate-wide view, so we can sort of detect if there were edge effects, corner effects for the knockdown failing to happen. And that's just one part of the Litichevskiy tool. We've also added, this is a static image, but we also have a Plotly version of the data, which is a little more interactive, where we're on the x-axis, we've listed the genes. These are over-plotted, but there's 330 here. But we've plotted the genes and we've sorted their rank, and so you can sort of see, like, if we scaled in here, you'd see these are all rank 1. All of these shRNAs work to knock down their gene. And then we start to get into the tail where things failed. And we can sort of zoom in since it's a Plotly app, we can zoom in and examine those in detail and see if there's anything we need to be concerned with, or if it's sort of just random failures. And another one we have is called QC Summarfy. Andrea Ekey worked on that. This aimed to, we had a version of this before, but she extended this and added a lot of new features. So basically the idea is to automatically capture and plot all of our QC measures and generate a weekly report of those. Involved, modification of the LIMS database, the SQL database, writing an ORM class in Python, report generation using Velocity, and then finally setting up a Flask app / API to be able to access it. And there's a few plots here that are static, but let me jump over to, here's the actual sort of live plots here, and you just see there's, I'm not going to go into a detail on a lot of these plots, but again, these are all really useful QC measures for the lab folk to assess the state of the assay, if there are any systematic problems occurring in lab over the course of the past week. Just jump down here again. We have plate-based views to see if there's any edge effects occurring or things like that. And again, it can be hard to tell with the heat map the extent of the color change. So there's another view of that with more of a bar graph, and this one's Plotly, so you can interact with it. Of course, you can zoom in and investigate as you need to see So for example, this is saying that well K22 had 19 failures across all the plates we ran. You know, you can sort of judge for yourself. You've got this long tail here. Well, this is sort of maybe a random failure rate of 10 wells. So maybe this is slightly above the threshold of what we should be concerned with. So let me just briefly give some more acknowledgements. I've tried to acknowledge people as I went. I'm sure I've missed people who I should have mentioned, but I would like to also mention Josh Gould does a lot of the app work. We do interact with him and get a lot of coding advice from him. And Jody and Jenny work on the knowledge base, and so does Arthur, and they're incredibly helpful for helping keep us honest and keeping things in reasonably good definitions. And then I want to also mention Maria and Andrea, Maria and Andrew and Bang for sort of making these really useful slides and graphics that go to making the presentation a lot easier. So thank you for your time, and I'm happy to take any questions.
The results from the multiple cell lines that are in the same well?
Yes, that goes to the core of Prism. Basically, the cell lines have been prepared so they have a barcode. And that originally what they did was they would sequence the material from the well and see how many barcodes were present. And then, so if a lot of barcodes were around, that means the cell hadn't been killed off. If the barcodes weren't around, it meant it had been killed off. And you could independently measure that. Now what we do is, it turns out the barcode is expressed as an mRNA, and we can detect that mRNA. And we actually do an experiment that's not the same as, but very close to the actual L1000 gene expression measurement. But instead of measuring actual biological genes, we measure the RNA version of the barcode sequence for the cell line.
I had a question about so my big takeaway from this is success from sort of thinking of computational biologists as people that can produce software that should become part of the engineering pipeline. So I was wondering if you could talk a little bit about how the lifespan of something, a project is created and how it gets folded back, like those QC metrics.
Yep.
Is there a production pipeline that those eventually have to get integrated into? And how does that handoff work? Like, how do you coordinate, someone who's not a software developer and not necessarily, not forcing them into a particular rigid model, but at the same time integrating it into something at the end?
Right. No, that's a great question. So so the life cycle sort of starts with, we have an idea either from the lab or from a manager saying, oh, we need to monitor. Let's say we're going to try to monitor X, or let me actually use this guy as an example, right? So we'd sort of done some of these plots in an ad hoc manner to sort of say, oh, we think there's a problem, and then we do it ad hoc, and we're like, all right, I'll make these by hand, I'll gather the data. And so generally what we do, we have that idea and we start working on it, so we set out to maybe write a really brief spec or just make sure we're all on the same page about what needs to get done, and then sort of turn the computational biologist loose on the problem, building basically a Python script that's going to, Python or MATLAB, but take some set of inputs, basically provide them with, this is the data you're going to have. Don't worry about how you're going to get that data, but this is the data that's going to be available to your script, and now start building these, generating these plots and things like that. That sort of takes me knowing that what data is available and where we're going to get it, and then later on, after we've sort of built the module, we then sort of work together to wire it up into the production pipeline. So it's sort of a combination of giving the person a well-defined sort of set of parameters and known inputs, and then having a system that we can sort of easily add the modules onto. Now, I will say that some of the work, especially the work Andrea did for this in particular, not the plotting part, but the separate sort of gathering plot, involved her actually scraping the file system and knowing where to look to sort of find the data. So that does come up sometimes as well. Does that answer? Okay, great.
That's it. Let's thank our speaker once again.
Thank you.